And welcome to our video presenting the project unveiling the secret of maternal nutrition of baby brain growth based on mass models.
This is a very interesting topic, but it's slightly complicated. So we decided to come up with a quick summary explaining the project by using interviews from staff and students. The students are exploring the impact of prenatal nutrition and maternal obesity on the fetal brain development by studying the structure of mice as order systems.
This experiment has been conducted in the Institute of Metabolic Sciences in the University of Cambridge, and interviewing Dr. Laura has showed us that this project holds great importance because they can help young mothers or women who are trying to conceive or get pregnant about the importance of being healthy during pregnancy to prevent passing obesity to their children. The students are hoping to gain a deeper understanding in the relationship between nutrition and brain growth.
Here is a demonstration of the experiment. Here is some clips of the interview we conducted with Dr. Laura.
A little bit what this experiment is about, what kind of equipment do you plan to use and everything, and what are the results that you're hoping to achieve and why this experiment?
Okay, so what we know from looking in human studies is that babies that are exposed to obesity and diabetes in pregnancy, when they grow up, they're more likely to become obese themselves. And because we know that obesity is caused by people eating too much food, we think that there might be a change in the way the baby's brain develops. So that's very hard for us to study in humans because we can't look inside human brains, but we're doing work in mice where we're looking if the mice mums have obesity and gestational diabetes in pregnancy, then we can look at how the parts of the baby's brain that control food intake develop differently. So the project that the students are looking at is that they've been mounting some brain sections from a mouse brain, and then later on we're going to identify the parts of the brain that are involved in controlling food intake and look at the specific number of some neurons called POMC neurons. And the special thing about POMC neurons is that they stop you from eating. So if you can switch on POMC neurons in someone's brain, then they will stop eating. And we think that maybe the offspring that have come from the obese mums have less of these neurons, and that's why they eat more, and that's why they develop obesity.
Yeah. So what is kind of like the genetic similarity between mice and humans? What is like their chromosome number or their percentage of similarity, etc., like this kind of stuff? Why did you actually decide to use mice?
So mice are like over 95% genetically similar to humans. They're very similar to humans in their genetics, particularly in the brain and the parts of the brain that we look at for controlling food intake. They are almost identical from mice to humans. So we know that we can look at things in the mouse brain and they would be the same in a human brain. Another really good thing about using mice in our studies, firstly, is that their pregnancies are short. So because we're interested in what happens in pregnancy.
So how long are their pregnancies?
3 weeks. A 3-week pregnancy. So we can't wait 9 months for a human baby to be born. So we have a 3-week pregnancy, and then the mice grow up quite quickly. So we can look at what happens to the mice when they grow up. Because once mice get to about 8 weeks of age, that's like being like a 30-year-old adult human. Another really good thing about mice is that we have a lot of genetically modified mouse models where you can turn on or off different neurons in their brain to control their feeding behavior. So we have lots of genetic tools that are available in mice more than other animal models.
One of the reasons that we all chose to discuss this experiment is our interest in metabolic sciences and to understand human complexity. We decided to show this in the form of a video in order to showcase these findings and evaluate the topic better. From the project, we learned the complexity of interacting risk factors and began to understand the problems that come with them more.
From the preparation of the tissue slices of mice brain using microtomes to
to mounting brain sections onto slides, staining, and using immunofluorescence, there is a lot of work to understand the complexities of maternal obesity and finding new and innovative ways to prevent it. Although this experiment does not contain a complete result, it helps us find out more about the relationship between these two risk factors of fetal brain development, nutrition, and maternal obesity.
We really enjoyed learning about this topic. Thank you for listening. ご清聴ありがとうございました。